Okay, people are still logging on, but I think we can get started. Welcome everybody to this week's Hutch at Home series. We're excited for this talk from Dr. Robert Bradley on Beyond the Central Dogma. And I'm Jeannie Chowning, a Senior Director of Science Education at Fred Hutch. And I'm just delighted that you're here with us today. And I would like to remind you that if you're not talking, especially if you're connecting via cell phone, to please mute yourself. As we go along, you can add your questions into the chat and we'll address them at a good break point or at the end. And we do have a feedback form at the end of the talk. If you could take a minute or two to fill that out, that gives us some good information for future sessions. Okay. So Dr. Bradley is a professor in both the public health sciences and the basic sciences division at Fred Hutch. And he's really working at the intersection of several different disciplines, including computational biology and biophysics. And he has been doing some fascinating work with RNA, and you'll hear more about that today. So I'm glad to turn it over to Dr. Bradley, and I will stop sharing my screen and switch it over to him. So there we go. Let me just make sure that you can share. Hang on just 1 second. Okay, should work now. There you go.
Great. Is it showing up okay?
It is.
Perfect.
We're seeing it. Oh, there you go. Yeah, very good.
Okay, great. Let's see. Okay, well, everybody, thanks for taking the time to come to the talk. I really appreciate it. I can't see the chat window, I think, while sharing my screen like this, or at least I don't know how to do that.
I can monitor for you.
That'd be great. Please don't hesitate to let me know if anybody's asked a question. So,
yes, we'll do.
Especially with the virtual nature, I feel like anything that makes it feel a little bit more interactive is a win.
Yeah, absolutely.
Okay, great. So I like to start all of my talks by thanking the scientists who actually made the discoveries and did the work that I'm going to talk about. I'm fortunate to have a really great group of students and postdocs in my lab, most of whose pictures are shown right here, as well as funding from both government agencies as well as private philanthropic foundations that have made all of our work possible. Before getting started on talking about the actual research that my lab does, I think it's really useful to think about the very big picture, the broadest perspective possible. So we are all familiar with the general concept that somehow genes are units of genetic information. This is a sentence that we've probably heard since high school. So what does this actually mean? Well, one way to think about it is in terms of a concrete example that feels intuitive. So if you take two dogs right here, they look very different. You might end up with this dog. This is actually my dog, whose name is Banjo. And he is sort of appropriate, I think, to show here because he's a hybrid. So he's a Labradoodle. So he's part poodle, part Labrador, 100% excellent. And he is a mix of the genes that he got from his two parents. And so as a result, he has some of the phenotypes, as we would say in science, or cute features, as I would say when I'm talking to him, of each of his parents. So what does this actually mean? Well, one way to actually conceptualize this in a way that's related to scientific concepts is thinking about the complexity of different organisms. And when I say complexity, I could mean a lot of different things. I could mean the repertoire of molecules that an organism could produce, or I could mean something like, what does it actually do that we experience daily? So if I think of corn, I think of tortillas, so it's really good. We can play this game where we think of other organisms. So this is a picture of yeast, which is famous for its ability to ferment. This is the aforementioned Banjo, who's really good at doing lots of things, including catching balls. And this, I'm ashamed to say, is me. Couldn't resist taking this picture when I was taking the photo of Banjo originally. And I'm a human, and humans can do lots of fancy things, right? So even Banjo can't compete with the suite of complex behaviors that a human can produce. So we know that genes are genetic information. So there's actually an old question in science, maybe 20 years old or more. And the question is, how many genes does every organism have? Now, we can answer this question in a straightforward way now using modern technologies. But 20 years ago, answering this question was not easy. And as a result, this is a question that both the popular press as well as the scientific community spend a lot of time thinking about. If we take these 4 organisms and order them by increasing complexity, thinking about which can do the fewest number of things and which can do the most number of things, we'd probably all agree that the order is something like this. That yeast is the least complex. Yeast can do amazing biochemical things, but it's not going to make a presentation in PowerPoint or Keynote. Humans, on the other hand, can do a lot of fancy things, right? We ourselves do a lot of amazing biochemistry, but we can also do really complicated things, like use the internet. So it would be natural to assume, therefore, that yeast would have perhaps a small number of genes. Corn probably would have more. My dog Banjo would have a lot. And humans would have a truly staggering number of genes based upon the complexity of organismal phenotypes that they exhibit. Or when I say organismal phenotypes, I essentially mean the repertoire of behavior that each organism can exhibit. It turns out that the answer is not what you might expect. So we now know that yeast has about 6,000 genes, which I'll tell you is a pretty big number. Corn has many more, so consistent with corn being a lot more complicated than yeast. But for Banjo, the picture's not what we thought. Banjo has a lot fewer genes than corn does, but I think Banjo is more complicated than corn. And I and you and all other humans somehow have about as many genes as dogs do, and we have way fewer than corn still. So this is very confusing and a little bit humbling. How is it that we, who can do things like wear a headset, only have something like 4 times as many genes as yeast? It's really weird. So I think we'd all agree that we can exhibit phenotypes that corn cannot. And we'd certainly all agree, without a doubt, that there's no comparison between what a human can do and what yeast can do. And yet somehow the number of genes we have doesn't reflect this incredibly complex behavior that we can exhibit. So just thinking of the biggest picture possible, we have this idea that genes are genetic information, right? I started this talk showing the example of my dog Banjo, who came from a Labrador and a Poodle. So somehow we know that genes are related to things about us. Am I tall? Am I not tall? Etc. Like, am I a human? Am I yeast? Somehow genes specify a lot of these things, and yet they don't exhibit a close relationship with what we intuitively experience in terms of organisms actually doing things in the world. So what's going on here? Well, in order to understand this, we actually have to get really detailed and think about the molecular biology of organisms and cells. So the concept of a gene is very abstract, but it turns out that the physical representation of a gene in a cell is actually very important, and it's critical to understand the way genes physically are represented within cells in order to understand how it is that we are so much more complex than yeast, even though we don't have a lot more genes. So these details are all going to connect to the bigger picture, if you'll stay with me here. So we know that we have DNA. DNA consists of 2 strands that wind around each other, shown right here, in what's called a right-handed helix. So this is the physical way that genes are stored in our DNA for every organism. And we know somehow that most of our bodies, that's at least the part of our body that's not water, is made up of protein. So protein is what actually lets us do things. It makes up our muscles, it makes up our brains, it makes up all of these different things. So this is just an example of the actual physical structure, molecular structure of a specific protein. So the question then is, how do we get a connection between DNA, which contains this abstract concept of a gene or genetic information, and then actually use it to make something that lets our bodies do something, namely make proteins? It turns out that even though genetic information is stored in DNA, that genetic information isn't used directly in order to make a protein. It first goes through an intermediary molecule called RNA. Now, as you might guess from the similarity between DNA and RNA, the acronyms are pretty similar. They're actually closely related molecules, but they're different. So if you look closely at DNA, you'll see there's 2 strands winding around each other, like I talked about. For RNA, it looks a little bit similar, but it turns out that RNA actually only has a single strand, but that single strand winds around itself. So RNA from far away kind of looks like DNA, but it's actually a very different molecule. It has very different chemical properties. This illustration that I've shown here, that DNA is used as the template to make RNA, which is used as the template to make protein, is a concept called the central dogma in molecular biology. And in many ways, it underlies most of what we understand about the molecular biology of the cell. The way that genes actually encode genetic information, and this is used to make proteins and actually execute molecular functions. So is RNA just this passive thing, or does it actually do something more? Well, I am an RNA biologist, and I will tell you that it actually does a lot of things. And one of the things that it does is it carries genetic messages both in a conceptual sense as well as in a physical sense. So virtually all of our DNA is located within the nucleus. So the nucleus of a cell in this illustration is the orange sphere. The cell itself is this kind of blobby, watery looking membrane on the outside. Protein is made outside of the nucleus in what's called the cytoplasm. So if the instructions to make proteins are actually in the nucleus, then how is it that protein is actually produced? Well, the way that it's actually produced is RNA is made in the nucleus, and then this RNA physically transports itself out to the cytoplasm, where it's used as the template to make proteins. So RNA is an actual physical messenger that carries genetic information within the cell. This gets us to ask how RNAs are actually made. Well, as I was just explaining, the RNA is actually produced in the nucleus. It exits the nucleus through a process called nuclear export, whereupon it chooses the template to make protein. All right, so this is what the central dogma is, but how does it actually connect to the picture that I was talking about earlier? Like, what does this have to do with the mystery of organismal complexity? Why we have only 3 or 4 times as many genes as yeast does, and yet humans are so much more complicated. The reason is that the process of making RNA is actually very complicated. A gene is not represented typically in our DNA as a single continuous stretch of DNA, all of which is used to make a protein. Instead, a typical gene in complex organisms like humans is broken up into a lot of different segments. So there'll be part of the DNA that codes for protein. This is part of the gene. And then there'll be a long stretch of DNA that doesn't code for protein. Then a little bit that codes for protein. Then a lot that doesn't code for protein, et cetera, et cetera. This is then made into RNA. So here I'm showing pictures of DNA. It's this double-stranded helix. It's made into RNA. So here you have a single-stranded molecule. So there's pieces of RNA that code for protein, interrupted by pieces of RNA that don't code for protein. This is all a single molecule. And then through a process called splicing, the pieces of RNA that don't code for protein are physically cut out, and then the pieces that do code for protein are stitched back together. This process is called Rna splicing. And the reason is because each fragment of RNA that codes for protein is spliced together, just like a long time ago you would splice a rope together. The reason this is related to the mystery of organismal complexity is the process is not deterministic. It doesn't always happen the same way. How do we know this? Well, one of the ways that we know this is that we can actually use incredibly powerful microscopes in order to physically see or visualize the process of RNA splicing happening. This is a famous micrograph or image from a microscope showing DNA. It's a little bit hard to illustrate this without a pointer, but DNA is the central thread running from the bottom to the top. And then there's RNA coming off like branches from a tree. And then the little loops at the end are pieces of RNA that are undergoing the splicing process, where the little loops are going to be excised and then kind of float away. This is what I mean when I say that the process of RNA splicing is not deterministic. You might end up with this image where you have a molecule that has the black segment, the red segment, and then the yellow segment. But you also might end up with a segment where the blue is swapped out for the red, and vice versa. And these things can keep happening in many different ways. So the picture here is that a single gene in the DNA is broken up into a lot of different segments. These segments have to be stitched back together in order to make an RNA that codes for a functional protein. But the way that stitching process happens, that RNA splicing process happens, can vary enormously. There's a really amazing example of the variability and incredible complexity of RNA splicing that comes from an organism that's much less sophisticated than we are. Drosophila, the fruit fly, has a gene called DSCAM. It's just one gene, and through the process of alternative RNA splicing, it can produce approximately 38,000 different proteins. So this is just a crazy example showing that in higher organisms, where I'm including organisms like fruit flies and higher organisms relative to something like yeast, this notion of one gene making one protein is no longer correct. It's vastly more complicated than that. So there's a total decoupling of our intuitive notion that one gene equals one unit of genetic information with the biological reality, which is that one gene can produce many, many different proteins, each of which can carry out a potentially different biological function within the cell. In organisms like humans or mice or the most complex organisms around, the vast majority of genes undergo this process of alternative splicing. The reason we now know this is due to a new technology called high throughput sequencing, which I'll explain a little bit more. But it enables us to accurately determine how many genes undergo alternative splicing. So in this estimate, which was 12 years ago now, we thought 86% of genes were alternatively spliced. And we now know that virtually every gene that is spliced in the human genome is alternatively spliced in some cell type or disease state. So we now have an answer to our original question. We only have 3 or 4 times as many genes as yeast, but the number of distinct proteins that we can produce is vastly different. In an organism like yeast, typically one gene makes one protein. This is what's most common. In an organism like human, is very different. We actually don't even know how many different proteins human cells can produce, but some people have estimated that perhaps human cells can make 500,000 different proteins. So there's this incredible decoupling between the number of genes and the numbers of proteins. As you can imagine, for a process that is so complex and intricate and fundamental to the notion of complexity of an organism, there's a lot of ways for splicing errors to occur. One disease in which splicing errors lie at the fundamental molecular basis of the disease is a muscular dystrophy called myotonic dystrophy. So this video shows one of the hallmark clinical presentations of the disease, where patients are unable to easily relax their muscles. So this patient can clench his fist, but not relax the muscle. More recently, we've discovered that errors in splicing underlie many different kinds of diseases, and in particular, cancers and diseases that are closely related to cancers called myelodysplastic syndromes. This is an image of specific types of healthy blood cells. If we take the same blood cells but cause an error in splicing, just a single error, then instead we get abnormal blood cells, where you can just see by comparing the top and bottom images that they visually look very different. This is not how the blood cells should look, and it causes this family of diseases called myelodysplastic syndromes. Another example is in a melanoma of the eye called uveal melanoma, where in this case, the cancer is caused by, again, a mutation that affects a single protein involved in the molecular process of RNA splicing, causing the melanoma. We now know that mutations that affect the RNA splicing process are very common in many different kinds of cancers, ranging from lung, liver, breast, pancreatic, melanoma, and many other kinds of diseases. The question, of course, is how is this happening, and what can we do about it? So there's been a huge revolution in our understanding of how important splicing is for human diseases very recently. Within the last 10 years, everything I'm showing you here in this slide has been discovered, with the exception of myotonic dystrophy. So 10 years ago, when I started my lab, we didn't know that all these errors in splicing were responsible for many different kinds of cancers, but now we do. And so this opens up a huge opportunity for us to understand why these cancers occur and how we can potentially treat them in the future. The problem, of course, is that splicing is a really intricate process and is very difficult to study. So we have a lot of fancy experimental tools that we can use to study molecular processes within cells, but they only sometimes work for splicing. This is an example. And from here on, I'm just going to say that I'm going to start talking about some actual data from my lab and from other labs. So it's going to get complicated. Please don't hesitate to ask questions in the chat or otherwise. If you have any at all, that would be great. We can use a technology called high throughput RNA sequencing in order to identify splicing changes that cause disease. So a typical question that we might want to answer is if we have a patient who has a cancer, for example, a breast cancer or a liver cancer, and we know that there's a change in splicing that's promoting the cancer or causing the cancer, why is that happening and how can we fix it? We can use this process called RNA sequencing, or RNA-seq for short, to start to answer that question. So if we have a patient who's willing to donate a sample from their cancer, as well as a sample from an adjacent healthy tissue, then we can perform this process of RNA-seq, where this is literally the machine that we can use, where we can take the sample that's donated by the patient, process it to extract the RNA, where RNA is the relevant molecule that we want to study, and then actually determine the identity of all the RNA molecules in each sample. These are what the data actually look like. Now these data are complicated, but what each group of 4 lines corresponds to, and I'll just say, I'm showing you the data because I think it's interesting to actually see like what it actually looks like what is the data that we're working with day to day. It's okay to not take away a lot beyond that. But what each group of 4 lines corresponds to is a single RNA molecule that was actually present in the cancer of a patient who donated a sample. So this is an actual RNA molecule, or groups of 4 of RNA molecules that we've seen in a patient. Now we can abstract away from this. One of the things that RNA-seq does is it produces a huge amount of data. So I just showed you a screenshot from my computer right there. But we don't just get data for 5 or 6 RNA molecules. We get data for hundreds of millions or even billions of RNA molecules for a single sample in a single patient. So it's so much data that is not even meaningful to actually visualize it like that. I showed you this image because it's interesting to see what the data looks like, but we never look at the data like that. There's so much that the human brain simply can't wrap its way around it. So what we do is we use statistical and computational tools to try to reduce the data to something that we as humans can actually understand. So if we take the same example of a patient who donated a sample from their cancer, we might reduce the data to something like this. Here is a really complicated plot, and what I'm showing you is RNA that we recovered from a cancer sample, as well as a healthy tissue taken from the same patient. And I'm showing you the frequency with which we observed RNA molecules arising from different parts of the genome. So at the bottom of this image is a single gene. Each gray box is an exon. So exons are the portion that code for protein. And then the little hat things are showing this process of RNA splicing. And in particular, the black exon is an exon that can be alternatively spliced. It can either be included or excluded. And if you compare just visually, you can see where I've highlighted in purple that there appears to be a difference between the cancer and healthy tissue. So if you look at the rest of the gene, it looks relatively similar. But if you look at that purple area, you see there's something really different. So there's a difference in splicing in this gene, which is called SRSF3 in this particular patient. Now, even this approach, which involved a lot of statistics and computation, isn't enough to actually let us work with the data, because although I told you we don't have many genes, we still have about 23,000 genes. So we can't look at 23,000 images like this for every single patient. It just wouldn't work. So we use further statistics and computation in order to convert images like this into a plot like this. So at this point, we're getting really complex and really abstract. Each dot in this plot corresponds to levels of a single RNA, either in the healthy tissue that we're analyzing, which is the x-axis, or in the corresponding cancerous tissue on the y-axis. The dots that I put in red are dots where we think that there is more of the RNA, and here I'm using this jargon isoform to refer to a specific RNA. An isoform is a particular outcome of the RNA splicing process. So a red dot corresponds to an RNA that's expressed at higher levels in the cancer, and a blue dot is one that's expressed at lower levels in the cancer, or equivalently higher levels in the healthy tissue. So in other words, the red and blue dots are RNAs that look different in the cancer. And so we're wondering, maybe there's something there that we need to investigate. So it's now straightforward to make these kinds of plots. And I want to note, here I wrote easy and put it in quotes. And the reason is because scientists are really fond of using words like straightforward and easy, when like what that really means is, you know, there's a protocol that I could write down that would be hundreds of pages, and after 10 years of training, like I'm ready to do it. But what these words, the reason I use these words is because they're really common, and you'll probably encounter them as you hear other scientists give talks, where really, you know, when I and other people say easy or straightforward, what we really mean is we know how to do it. It actually doesn't mean it's easy at all. Typically, it means it's difficult,
Rob, we
but we
have
know
a
how to do it.
question in the
Yes,
chat, and I think this is referring specifically to this slide. Is this for one person or multiple people with, say, liver cancer?
that's a great question. So this image, the question is this for one person, or is this image representing data averaged over many patients? This is actually just for one person. So the person who asked that question perhaps might be wondering, what if we want to look at a whole cohort of patients? What if we want to study what's happening for 50 or 500 or 5,000 individuals? And that's actually something that we do a lot. And so the reality is that we need even further statistics and computation to start to approach that question. So how do we take a plot like this and make it meaningful for 500 people? And we have good statistical ways to do this as well. So we can make plots like this, but in many ways, they don't tell us what we really want to know. What we really want to know is which are the specific RNAs that actually matter. So how do we make statements that are actually important for patient outcomes or for the development of new therapeutics? So we're really good at characterizing what's happening. With modern tools, and by modern, I mean very recent, in the last 5 years or maybe 10 years, we've developed the ability to make a lot of descriptions. So we can take a cancer in a normal tissue and we can identify a lot of what appears to be different. But we don't know which of those differences are actually important. And the reason that matters is because if we want to design a therapy, if we can say there's 5,000 differences, but we don't know which one to target with the therapy, then we don't know what to do next. And so a lot of the effort in my research lab is focused on determining how we can actually make biological statements about relative importance of changes. So if we take a plot like this, how do we know which of these circled dots is the most important? There have been a lot of breakthroughs in answering the question of relative importance and biological function of individual genes. There's a molecular process called RNA interference, or RNAi, as well as more recently, the idea of a CRISPR-Cas gene knockout. So you've probably heard of CRISPR. And both of these techniques offer a way to take a gene and essentially prevent it from functioning normally. And we can use that ability in order to identify or determine what role a given gene is playing in a cell. But unfortunately, we can't use that same technology in order to identify the role that a given isoform or messenger RNA is playing within the cell. The reason is because each of these technologies suppresses the production of RNA from a gene. It doesn't just affect a single isoform resulting from alternative splicing. So this is a really complicated idea, but the basic idea is that if a gene can produce 10 different forms of RNAs, we're really good at suppressing all of those RNAs at once, but we're not good at suppressing just a single one.
Rob, there's a question. Can you look which ones happen first? And I think this might have been referring to just where you were before this slide.
That's a great question. So if the question, I think the question is about the order
Sequence.
in which things happen.
Yeah. Mm-hmm.
The short answer is no. So I think the question might be getting at if we see 1,000 different RNAs that are what we call aberrantly expressed, that are present at higher or lower levels than in normal tissue in a cancer, can we identify what happens first? That's a good idea. But unfortunately, we typically can't do that. And the reason is because we don't have what's called longitudinal data. So this is data that follows a cancer over time. And the reason is because typically cancers are detected, they're not detected at the very early stages. Like a cancer is virtually never detected when it's not yet a cancer. It's only detected after it's actually, or at least almost always only detected after it's become an overt cancer and becomes something that either it causes symptoms or can be detected with modern screening tools. So the reality is that many cancers probably form when they're very, very small, long before we ever are aware of them. And so we unfortunately just don't have a way to detect those or perform sort of research level molecular profiling that would let us identify what changes occur first. But it's a great idea. So the broad picture of where we are now is that thanks to this revolution brought about by the technology called RNA sequencing, we're really good at making descriptive statements. So we're really good at identifying lots of different RNA molecules in cancers, for example. So this is a cartoon that a postdoc in my lab, James Thomas, drew. So the solid gray line has increased dramatically. But the dashed gray line, which is what we actually want to know, what the biological function of a given RNA is, has not increased accordingly. So we're really good at making scatter plots like I just showed, or coming up with a giant table that you could bring up in Excel of all the changes that occur, but we're not good at saying these are the 4 that actually matter. So that's a really tough question. So my lab has been working to address this problem by developing new tools that are based on the CRISPR technology. And in principle, it's very straightforward. So here I'm showing a cartoon of 3 exons. You can imagine that this is a gene, and this gene can produce 2 different isoforms or 2 different mRNAs. So one mRNA might consist of the 2 exons on the outside, the first and the last. The other mRNA or messenger RNA might contain all 3 exons altogether. So we can use CRISPR to physically cut out the center exon, the center piece of DNA that codes for protein. So now the gene looks like this. And because of that, this modified gene, after we've delivered our CRISPR technology, is only capable of producing the RNA shown on the bottom right and not the RNA shown on the bottom left. So this gene can produce 2 different isoforms normally. One has 3 exons and one has 2 exons. And again, please remember, I'm using isoform and RNA interchangeably. I know that's a little confusing. And we can use CRISPR to change the gene structure so that it can only produce the isoform with 2 exons. So this offers a way for us to actually begin manipulating what RNAs a cell makes without destroying the entire gene. So typical ways of using CRISPR just destroy the whole gene. And we're trying instead to just change the RNAs the gene can make. We can use this technology to do all kinds of things to cells. So here's an example where there's a protein, and the protein is tagged orange in this picture. This protein normally accumulates in the nucleus of cells. This is a protein called muscleblind. So normally it's like present in the nucleus in a lot of cells, and you can see it in bright orange reflected here. We can use our CRISPR technology to delete a single exon in the muscleblind protein, and the single exon codes for what's known as a nuclear localization signal. So this is a special piece of protein sequence that tells the cell this protein belongs in the nucleus. So we use our CRISPR technology to change the gene structure so that the muscleblind gene can no longer make the mRNA or the messenger RNA or isoform that goes to the nucleus. And then we end up with this picture. So it's much more diffuse. There's still some cells that have the muscleblind protein in the nucleus, but many more have it sort of diffused throughout the whole cell, including in the cytoplasm outside of the nucleus. We can test whether normal splicing of a gene is critical for cell survival. So this is just an image of cells growing in a petri dish in cell culture, as we say. You can see that they're healthy. These are cells that are not healthy. These are the exact same cells, but what we did is we got rid of one of the RNAs produced by a gene called U2AF1. So we used our CRISPR tool in order to delete an exon from the gene U2AF1, which is actually a gene that's involved in the RNA splicing process itself. And then we saw that the cells died extremely rapidly. So the point here is that the RNA that we deleted from these cells appears to be absolutely essential for cells to survive. They can no longer grow. They have this strange morphology or strange shape. We can also do really complicated things, as reflected by really complex data in this slide. Here, what we did is we used our CRISPR tool to target lots of different genes. Each gene is represented by an uppercase italicized acronym. So the top one, for example, is HNRNP-A2B1. Most human genes have these terrible names that are impossible to remember. HNRNPD, HNRNP-H1, et cetera, et cetera. So each of these is a gene. And then we took our CRISPR technology, took lung cancer cells for each of these genes, used it to delete a single RNA produced by each of these genes, and then tested whether or not the lung cancers could still grow well. And what we saw in this case is that deleting the RNA that we targeted for each of these genes prevented the lung cancers from growing effectively. So we would say that each of the RNAs that we targeted is essential for tumor growth, represented by blue in this diagram. We did something interesting, which is we targeted a different RNA produced by the exact same genes. So if you look at HNRNP-H1, for example, that's the third from the top row on the left. You can see it's blue. So getting rid of that RNA is bad for tumor growth. We would say that RNA is essential for tumors to grow. But in contrast, when we looked at some other RNAs made by this exact same gene, HNRNP-H1, in this case shown in the top two rows on the right, you can see that getting rid of one of those RNAs actually is very good for the tumors. They grow much more quickly. So we call an RNA like that tumor suppressive. It's an RNA which, if present in a cell, makes it difficult for a tumor to grow. So when we destroy the cell's ability to make the RNA, the resulting cancer grows much more quickly. And what's really interesting about these data is you can see that there's a lot of blue on the left and a lot of red on the right. So depending upon which type of RNA we get rid of in the same genes, we can either make tumors grow slower or grow more quickly. What's really exciting about this new tool that we developed that's based on CRISPR for manipulating which RNAs a cell produced is that we can get insight into things that actually matter for patients with cancer. So everything that I just showed you before is using very abstract models where we're taking cells and we're forcing them to either make an RNA or not make an RNA and then seeing how it affects the growth of tumors. There's no guarantee that that's directly related to what actually happens when a human patient has cancer. And so in order to test that, we used our assay to identify RNAs that we thought promoted cancer growth versus those that slowed cancer growth. We then took data from samples that were donated by about 500 patients with lung cancer, and we categorized those samples based upon whether they contained low versus high levels of RNAs that in our assay we thought promoted or repressed cancer growth. And we found that when we stratified patients like that, we saw a big difference in the fraction or the survival of patients with or without high or low levels of these RNAs produced by alternative splicing identified in our assay. So the big picture is that when we started this particular project a couple of years ago, we didn't even have a way to manipulate which RNAs a cell was producing effectively. Now we have a way to do that, and we can use this new technology in order to identify things that actually appear to be quite important for the growth of human cancers. So we can further combine these technologies with statistical approaches in order to actually create new therapies. This is what the data actually looks like, and this gets to one of the first questions, which is, how do you handle the case of not just one patient, but a case where perhaps thousands of patients have donated samples? In this project that I'm just sort of showing a very high-level outline right here, we had data from thousands of patients with many different types of cancers. So we put this all through the RNA-seq assay using that machine that I showed you earlier. This generates a huge number of data points, so many that it's hard to even conceptualize what that means. And then we can iteratively apply different computational or experimental procedures in order to reduce these data to something that's more meaningful. We can take all the raw data and then make actual statements about estimates of abundance of different types of molecules. We can then average across lots of different patients with lots of different types of cancers to find molecular changes that are unifying across patients. So the ones that we think are the most likely to be important. We can then use our new CRISPR tool in order to actually find the molecular changes that we think are the most important for driving the cancer's growth. And then of those, pick one that we think is a good therapeutic target and actually create a new targeted therapy. Where in this case, we were able to identify a compound that reversed the splicing change to slow the growth of melanomas. Ultimately, we want to use these approaches in order to design what we would call precision medicine therapies. So therapies that are actually tailored to the specific molecular changes that cause a given cancer type. And here's an example from a study that we published a couple of years ago, where there's a gene called SRSF2 that is commonly mutated, and when mutated, it can cause leukemias. In this example, we took 2 leukemias that either did or did not have an SRSF2 mutation, and these 2 leukemias were otherwise completely identical. This is the burden that we saw in our model system for these leukemias. So these leukemias were very aggressive. When we treated them with a compound that we identified as potentially therapeutic, we saw no effect in leukemias lacking a mutation in the gene SRSF2. In contrast, we saw this big therapeutic effect, specifically in the case of an SRSF2 mutant leukemia. So the broad idea here is that by understanding the basic science and molecular biology of cancers, using these new technologies that we've talked about, we can actually create therapies that appear to have big effects in a way that's conditioned upon the actual genetic structure of the disease. All right, thank you all for listening, and if there are any other questions, I'd love to take them.
That's great. Thank you so much. Please feel free to put your questions into the chat, or at this point, you can also choose to unmute yourself if you prefer. I was wondering are there commonalities among the types of splicing leading to cancer? Are you finding patterns there? And that kind of led me to another question about, you mentioned that sometimes there's errors in the splicing machinery and how often those are associated with cancers.
Yeah, those are really great questions. There are mutations that affect protein components of the splicing machinery that affect 4 specific proteins. The jargon, or their names, is SF3B1, SRSF2, U2AF1, and ZRSR2. And what's very interesting and surprising about those 4 genes is that before they were discovered about 10 years ago, nobody would have guessed that those genes, when mutated, would cause cancer. And the reason is because all of those are genes which are expressed in every cell and thought to be absolutely required for cell survival. So if you take, for example, a cell and just eliminate one of those genes, then that cell is unable to survive, typically. But in very specific contexts, if you, instead of eliminating that gene, just create a subtle mutation that changes the way that it participates in the RNA splicing process, the cell somehow is able to become tumorigenic. So it was very surprising, and it's really been a mystery trying to figure out why these specific genes are so important for cancer. There's not really a commonality, to get to your other question, between these 4 genes that would lead to them, that would let us have guessed ahead of time why they were going to be the critical ones. That's another really interesting, mysterious question. So we and other people are working hard to try to figure it out. But so far, the picture seems to be that you have to study every single gene and protein that's mutated in order to understand how it's causing cancer.
Thank you. There was more of a comment than a question of somebody saying thank you, and that did blow this person's mind a little, because they didn't realize that there could be alternative RNA splicing. So thank you for expanding our knowledge on that. Other questions that folks might have? I invite you to contribute.
This is more of a question about the process. You talked about how in one of your graphs, it was the data for 1 human, but then when you look at populations of people, what is the process like for cleaning that up? Does it take a lot of time to look at a lot of people?
That's a great question. It does. It does take a lot more work to try to understand what's happening in a large cohort of patients rather than just in one patient. But there's been a real revolution in the last few years in, I would say from the clinical perspective, the ability to assemble large cohorts. It takes a huge amount of work from doctors, and it takes a really engaged and generous patient population to participate in these studies. And as a result, we now have data sets that we couldn't have dreamed of 10 years ago that are letting us really make just a totally different level of insight into the disease process. there have also been revolutions in our ability to computationally analyze these data, to the point that in many ways it actually can be almost easier to work with a cohort of many patients than just a few. And one way you can think about it is, we all know, I think, or at least it is frequently talked about, the fact that each cancer is a little bit different. This is one of the reasons that cancer is so tough to understand and treat. It's not as if two cases of breast cancer are the same. And so one advantage to looking at a large cohort is that because each case of breast cancer or prostate cancer or liver cancer can be a little bit different or even very different from each other, when we compare them all together, any changes that remain in common have a high likelihood of being very important.
For a lot of our speakers, we've been asking them a little bit about their journey to their science career. Could you talk just briefly about, kind of how you got interested in science, where you grew up, what was high school like, how did you end up where you are now? Just, I think that's of interest to this audience.
Yeah, definitely. I'd love to talk about it. So I grew up in Birmingham, Alabama, a long time ago. Lived there till I went to college. And the way I actually came about being a scientist is I took physics for the first time in 10th grade, and I thought it was just the most amazing thing ever. I remember being in high school and this idea that you were supposed to figure things out and it wasn't about memorization really appealed to me. So I thought that was really amazing. And I have to say, I'm a biologist now, and in high school at the time, I did not find the way biology was taught to be especially appealing. So when I was in high school, at least, it was more about memorization than about figuring things out, and that didn't appeal to me very much. So I actually decided very early on I was going to be a physicist, and kind of made all my decisions based on that. Like, I chose a college based on wanting to major in physics, and then after college, I chose to go to a specific graduate program because I wanted to work with a specific Ph.D. advisor who was a physicist. And so I made all these life decisions. And then I actually was at a physics conference when I was in grad school, and just all of a sudden realized this, in hindsight, very obvious thing, which was one of the reasons I wanted to be in physics, was that I felt like if you looked at the modern world, physics had kind of defined it, right? Like, physicists created the internet, they created lasers, computers, atomic energy, like all these things came straight from physics. And I wanted to be part of this grand endeavor, but I kind of had this realization that maybe this wasn't going to be true for the 21st century. Making this decision based on the past. So I was literally at this conference when I kind of had this realization, and then I started brainstorming on this piece of paper about, so what does this mean? Like, what should I do? And one of the things that I wrote down, two things that I thought were going to be important in the 21st century. One was computer science, and one was biology. So I started trying to learn about those from that day forward. Left physics, and kind of had a long journey to get where I am since then. Like, I was in grad school at UC Berkeley, and I went through 4 different departments at Berkeley. I was in physics, and then biophysics, then math and molecular biology. So I had a very unusual path, I think, to get to be where I am.
Do you think that kind of background, though, has aided you in the work that you do now, to have had that broad exposure to different disciplines?
I think, I'd like to think so. I think it definitely comes with some advantages. So one is, I think, just having seen a lot of different disciplines and tried to enter new fields gives, I think one pro is that it can give you confidence that you can figure something new out. Figuring biology out as an adult was very tricky. Like, I haven't taken a biology class since 11th grade. Wow. And so it's been sort of, this long-term struggle for me to try to rectify that. So that's been really good. I think one thing that I started off by, I think, a little bit criticizing physics, which is not certainly what I meant to do, because it's the reason I'm a scientist. But I think one thing that is truly amazing about physics education is that it's very focused on problem solving. The idea that you define a question, then you answer it. And I think that's something that's an approach that has really served me well, not just in my professional life, but just my whole life in general.
That's great. Thank you. Other audience questions? What are you finding out about the same RNA splicing issues across different types of cancers? Will driver mutations connect prostate, breast, lung cancer patients instead of their type of cancer?
That's a really great question. And it gets to an issue that I was talking about a little bit earlier, which is that if two patients have breast cancer or liver cancer, that doesn't actually mean that the cancer cells are similar to each other. And one reason is that we now know that cancer is a genetic disease that is caused by genetic changes. And so as the questioner was highlighting, it turns out that many different genes, including the splicing factors that my lab focuses on, are recurrently mutated in lots of different cancer types. So one thing that we found to answer the question is highly similar splicing changes, splicing errors in almost every kind of cancer that has a splicing factor mutation, that has a mutation of the types that I've talked about in my presentation. So if we look, for example, at a breast cancer with the mutation in the splicing factor gene SF3B1 and a melanoma with that same mutation, they look much more similar to each other than they do to, say, a breast cancer cell without that mutation. So one really exciting thing about this is it offers a way to immediately translate any therapeutic developments that we can come about to other cancer types.
Good. I was wondering, what's the distribution of like splicing frequency across the genome? Like, are there some like hyperspliced genes and some, you know, that hardly ever splice? And
That's a great question. Yeah, the short answer is yes. This is actually something that's not very well studied, but I've always wanted to study it.
yeah.
So there's kind of a related question is there's a small set of genes that are never spliced, and it's kind of confusing because we now know that the process of splicing itself is actually really important for most genes. If you take a gene with an intron, the intron being the part of the DNA that's not used to code for protein, and you get rid of that intron, then typically the gene doesn't work very well anymore. So if you get rid of this thing that doesn't seem to matter, you find out it actually matters a lot. And so it's very mysterious thinking about why some genes are not spliced at all. And some genes, just like you said, have an incredible amount of splicing. And we don't really know why in most cases. So I think it's a very good and deep question, but one to which I can only speculate as to answers.
Great, thank you. Okay. If there are any other questions, feel free to unmute yourself or pop something in the chat. Could you stop sharing your screen? I'm going to switch over to my last few slides here. Thank you. And somebody also wrote, "Boo for the biology classes that only lecture." So, you know, it is true that, you know, there's something that I think a lot of us biology educators think about is how we can incorporate more of that spirit of figuring things out into biology class. And so thank you for that. Okay, let's see, share my screen. Okay.
Hey, I guess I have one question. Thank you, that's a great talk. I'm just wondering if, so you have these, what I assume is probably point mutations in your splicing factors, since they don't knock them out entirely, but they change them. Does that, how many transcripts does that affect? Or is it very specific? Like if you look at the effect of the splicing factor, are they only like, how specific are the defects?
That's a great question. So just to expand a little bit for those who aren't familiar with the details of the biology, so the question is, many of the mutations that I was talking about affect proteins that actually catalyze the RNA splicing process itself. And because almost all genes are affected by splicing, does this mean that every gene is messed up in these cells, or does it mean that only a tiny number are messed up? What happens is it's kind of in between. It's almost exactly the wrong scenario in terms of figuring things out. So for a typical splicing factor mutation, we'll see a few hundred genes to maybe a few thousand affected. And the reason I say it's exactly the wrong number is because if only a tiny number were affected, say 5 or 20, then we could just test them all and we'd know all the answers. If every gene was messed up, then we could almost throw up our hands and say, well, it's causing such complex changes that there's no hope of understanding it. But because it affects an intermediate number, we are in a scenario where we have the hope of figuring out which matter and which don't, but it's very hard to do so. And so that is the ultimate motivation for a lot of the technologies that my lab is developing and that I was telling you about, to find ways so that we can test hundreds or thousands of changes very rapidly to find which ones are most important.
And I wonder, so is that set of 10 to hundreds or whatever it is, that's the same for every mutation of splicing factor you have, or what's the overlap?
That's also a great question. So several different splicing factors are recurrently mutated in cancers. The overlap is actually extremely small. So one, I think, intuitive explanation for why these four are mutated is exactly like you're suggesting, that perhaps they converge on a small set of genes that cause the cancers. But that doesn't appear to be the case, unfortunately, because that would be very intuitive and it would very much make sense. But unfortunately, it doesn't, as far as we've seen, does not appear to be the case. It seems like each splicing factor mutation is very different from all the others.
Wow. Thank you. Thank you very much. I have up the SEP News and Events page, which posts all the upcoming Hutch at Home events, as well as recordings of past events. And so this recording will be up in a day or two. And so you can send these out to students and revisit some of the talks if you like as well. And I just wanted to mention, we're not going to have one July 7th, but we will continue through the summer and maybe into the fall. Our next speaker next week is Ricky Padilla de Valle, and he's going to be talking about transposons. And he's a molecular and cellular biology graduate student. He's at the Pacific Northwest Research Institute, but he's at the Fred Hutch UW graduate program. So looking forward to hopefully seeing you there. And then just remind you about our feedback form. A lot of people are having trouble with the bit.ly. The last letter there, if you're not hyperlinking, it's pasted into the chat, so you can hyperlink if you like. But if you're entering it in, that last letter is a lowercase l. You can also use your phone to scan the QR code. So if everybody could express your appreciation either in the chat window or unmute yourself or throw up a little icon for Dr. Bradley's talk. Thank you so much for joining us today. There's a number of people have put in things into the chat with their appreciation. So thank you for joining us. It was very, very interesting.